For our first talk, our first speaker is Dr. Anabel Flores from California Baptist University. She's going to be telling us about her work, the work in her lab on host pathogen interactions between the innate immune system and Listeria and Salmonella.
All right. Hi, everyone. I am so happy to be here, and I hope that you've been enjoying this conference as much as I have been as well. I'm going to stand on this side because I tend to disappear when I stand behind the podium. So I'm going to be on this side. You know, you guys just had your lunch, wonderful lunch, by the way. And now we're going to go ahead and talk about foodborne pathogens, something like Listeria. So I'm an early career faculty member at California Baptist University. California Baptist University is in Riverside. If you've never heard of Riverside, that's in Southern California between Los Angeles and San Diego. It is a primarily undergraduate institution and a minority serving institution as well. And the proposal that I'm going to talk about here today is for the NSF Building Research Capacity of New Faculty in Biology grant. And I think CBU is such a great institution for this type of grant where we could start to expand our research capacity. And because, you know, grants go through a run of revisions, this is my new working title. So I've actually taken both Listeria and Salmonella into two distinct projects and two distinct grants. So my new working title is Exploring Listeria Host Macrophage Interactions Under External Antibiotic Stressors. All right, so brief outline, you know, I'm going to take you through a brief background, M1, M2, and broader impacts. You know when I'm done. All right, so a little bit of background. Bacteria has been here since the beginning of time, right? It exists in many shapes or forms. There's the spheres, rods, spirals. Bacteria could be very beneficial, like it's in the case of soil fertility. If you have a really good mix of bacteria, then usually you see an increase in things like crops, in agriculture, and things like that. It could even be beneficial to the host, right? Like it is in the case in mammals. We have a lot of really good bacteria in our microbiome and all of that. But that is not always the case, which is in the case with Listeria. So Listeria is a gram-positive foodborne pathogen, and it is one of the two strains of this family that is capable of infecting humans. It is distributed widely in nature. It is found in the soil, vegetation, and even in the stool of mammals. And because of this, it's also actually has adapted, it's very versatile, it's able to adapt to different pH, so it's able to survive within the digestive tract of, you know, mammals. And it's also very flexible when it comes to temperatures. It is able to withstand very cold temperatures. Makes it a little bit tricky, right? And due to this, Listeria outbreaks are quite common. I am pretty sure you've all heard about these outbreaks in the news, right? It's almost one every month or something like this. However, while Listeria outbreaks are quite common, the Listeria infections, systemic infections, are sporadic. And this may be due to the fact that in mammals we have very good, healthy mammals, I should say, very good defense mechanisms, right? In humans specifically, we have two different types of defense mechanisms. We have innate immunity, crash course in immunity right now. So we have innate immunity, which is rapid and not selective. We also have adaptive immunity, which is a little bit slower, but it's more selective. I'm going to focus in innate immunity. So here you have a lot of different types of cells, mast cell, dendritic cell, but I'm very interested in macrophages. That's my favorite type of cell. So macrophages are in type of cell for innate immune response, and it's sort of the first line of response when host encounters a pathogen. There's two different, well, for simplicity, there's two different flavors of macrophages, M1 and M2 macrophages. The M1 is simplified as the killing phenotype, while the M2 is the healing phenotype. So for the M1 phenotype, when this macrophage encounters a pathogen, something like a bacteria, there's different ways that they will try to get rid of this pathogen. So first, phagocytosis, uptake of the pathogen. Then you have the production of reactive oxygen species. And then you have extracellular traps, right? Different flavors. But sometimes it is not as easy to get rid of a pathogen, right? Again, coming back to Listeria. Listeria, I like to think about it as a Trojan horse of bacteria. It's very interesting what they do. They're very promiscuous. So when it encounters a macrophage will intake it via phagocytosis to try to get rid of it. Once inside this phagosome, what does it do? It enlists this sophisticated arsenal, and it's able to break out of the phagosome and go into the cytosol of the macrophage. And so once in the cytosol, it is able to proliferate, replicate, go infect other cells, and then you're in trouble or we're in trouble, right? All right, so the intracellular listeria is very difficult to eradicate. And this may be due because when you have a bacterial infection, what's the first line of treatment? Antibiotics, right? However, there's some research that supports the idea that antibiotics actually serve as signals or cues to these bacteria so that they're able to change the phenotype of the bacteria. So in a sense, you're rearranging their genetic makeup and their phenotype to make them more resistant to antibiotics. So the ability to remodel and avoid susceptibility to external stressors, such as antibiotics, is gaining quite a little bit of traction in Listeria. While it is not the bacteria that has the most antibiotic resistance, it is on the rise. So that's a type of concern. And that's what makes Listeria a very attractive pathogen and very interesting. A lot of people study Listeria. It is widely studied how it infects the host and how it the infection progresses. What is not widely studied is how the host macrophage is remodeled by external stressors such as antibiotics, and that's something that I'm very interested in. So to this date, the role of antibiotics in modulating macrophage response during bacterial infection and progression remains unexplored. And so the proposed research project that I have here, which is for a research program at California Baptist University, seeks to address this knowledge gap by exploring Listeria host macrophage interactions under external antibiotic stressors using THP1 macrophages. So my first aim is to evaluate the intracellular killing of Listeria by macrophages in the presence and absence of antibiotics. And I propose to do by first beginning to do a screen in which I will screen different strains of Listeria, because you have very different variations of that, against 3 different antibiotics that I've chosen due to previous work and my postdoctoral work. So these 3 different antibiotics, ampicillin, ceftriaxone, and dapsomycin. And I propose to do this by using what we call minimum inhibitory assays, which is a antimicrobial susceptibility test standard in the microbiology labs. And so in this test, you're testing different concentrations of antibiotics against a bacteria, and you're able to determine the lowest concentration that inhibits the growth of this bacteria. I also propose to do time kill assays in which here you're determining the lowest concentration of antibiotic needed to inhibit bacterial growth, but it's not really telling you at what time point it works. So here I propose to do the time killing, which we will look at different time points and able to determine where the antibiotic is most efficient. Then for the sub-aim, I propose to look at the macrophage viability by testing on some of those different ways of killing the macrophages that I already introduced to you, such as phagocytosis, reactive oxygen species, and extracellular traps. And this will allow me to determine the best candidate strains and the concentrations of antibiotics to use for my aim-2. So for aim-2, I propose to look at long-term exposure and short-term exposures to antibiotics and cytokine release in macrophages by doing ELISAs and looking at different inflammatory and anti-inflammatory cytokines, as well as PCR. And again, this is just to determine the best candidate strains and the best antibiotics that I would use for doing looking at dual RNA sequencing. And this is to examine how Listeria and antibiotics shape the host macrophages gene expression and level variation. And this is a fairly new technique which allows you to simultaneously look at transcriptomics of both the host and the pathogen at the same time. So that would be really interesting to look at. And this will allow us to identify several candidate genes for future work. So I am very excited for that. Previous work from our collaborators has shown that when cells are, when Listeria is pre-exposed to antibiotics overnight, they become more sensitive to neutrophil killing, which is shown here. This is the untreated, these are the treated options. And what's interesting is when you do the exact same experiment with whole blood, you see an even bigger reduction in bacterial killing, suggesting that there's other different innate immune molecules involved in this killing or getting rid of this Listeria pathogen. And okay, I think that macrophages play a big role in this, which is why I'm building up on this previous research and building my research and looking at macrophages. Preliminary work that has been done at CBU, I've established a BSO2 research lab, very, very hard. I've also established an institutional biosafety community at CBU. My students have been working really hard to establish a THP1 cell line and looking at different types of cell viability assays, such as visualizing phagocytosis and all of that, which is really interesting. And as part of this grant, you're allowed to propose the broader impacts of these grants. So I proposed to build a pipeline in which we're involving a local high school, Indio High School, and this is where they have a high percentage of first generation college students and minority groups. We currently have an ongoing collaboration with them. So I propose to work with them in which we involve them early on in STEM projects, small projects which they could collaborate with students at CBU. I'm proposing to also build a cure course at CBU in which it would expose undergraduate students early on to research projects. They're working in collaboration with one another. Their findings would be presented at an annual research conference at CBU. And also this cure course will serve as a pipeline or a gateway to train students in the techniques that would be used in research that I'm proposing to do here. Because Listeria is a BSO2 agent, safety is of concern. So this would be a really good course to train them how to properly work with bacteria so that they are able to do it with that BSL2 agent. And with that, I'd like to acknowledge my institution, California Baptist University Internal Research Grant, which supported a lot of this preliminary work and building the lab from scratch, which is a lot of work. My enthusiastic students, these are my students from last year, very enthusiastic and optimistic. When things didn't work, they always kept the lab going. And if you've ever built a lab from scratch, it takes a lot of work, and they've just been so great at doing that. My postdoctoral mentors, Monica Kumaraswamy and Victor Nisset at UCSD, for helping me develop this research project, and Brown University for hosting this amazing conference that we've all enjoyed, I'm sure. And Allison and the organizing committee, Kimberly, Miles, wherever you are, thank you so much for the great amount of work that you guys have put into this conference. And with that, I'd be happy to take any questions.
So once the bacteria is within the phagosome, do you know what kind of the signaling events that allow it to leave the phagosome and not be, degraded or killed?
Yeah. So there's different types of signaling pathways that are involved. So it's not just one, but depending on which bacteria strain you're talking about, they enlist breaking of the permeable membrane in the phagosome to break out and essentially get out of the phagosome and go replicate itself in the cytosol. But yeah, very different pathways, and because not every single strain of bacteria is exactly the same, they've all kind of adapted different types of techniques or different types of arsenal to get out of the phagosome.
I guess I had a similar question. First of all, that it's amazing that you're pushing through and setting this up at a predominantly undergrad minority serving institute. As far as like the macrophage story, so do you think that the antibiotics are working directly to I guess give cues to the bug, or is it also synergistically impacting the ability of macrophages to to switch from anti-inflammatory to pro-inflammatory? And are you considering looking at mitochondria? If you are, we should collaborate.
We should. And yes, absolutely great question. My hypothesis is that the antibiotics are pushing it to a more M2 phenotype. And because we've seen this in other different types of conditions like cancer, a lot of work has been done in cancer with this type of macrophage. So I do think it's pushing it into an M2 phenotype. So that's why I'm interested in looking at the different anti-inflammatory cytokines. what I would also be interested in looking at is whether it's reversible, right? Because there's this thought that if the antibiotics are pushing the macrophages to the M2 phenotype, is it also reversible? And when you reverse that phenotype, is it also going to affect antibiotic resistance, right? So if macrophages essentially are also a factor in antibiotic resistance. So that would be interesting to look. And yes, we should talk.
Do we have time for one more question? No. Okay. So I think thank you so much, Anabel, for that wonderful presentation.